Most traits or phenotypes are not controlled by a single gene, but in fact by many genes. Discovering all the genes that control a particular trait and also how those genes interact to determine the phenotype is an important goal in genetics. And geneticists use an algorithm for discovering genes and their interactions. So the first step in this algorithm is to obtain many strains, lines, types of organisms that are mutant for a particular trait. Now, you could go and identify rare but naturally occurring mutant individuals and breed them in the lab, so you can find mutants that are naturally occurring, or you can also generate mutants by mutagenizing individuals using X-rays or ethyl methanosulfate or EMS. Once you have established different strains or lines that are mutant for the same trait, then the next goal is to determine whether, if you pick two lines, is it the same gene that has been mutated in both the lines, or is it that two different genes have been mutated in those lines? And once you have determined whether it's the same gene, so two alleles of the same gene, or whether it's different genes, then you can work out how those genes interact by analyzing double mutants. Let us discuss the second step, which is determining whether in a pair of lines or strains the same gene has been mutated, or it's actually different genes that are controlling the same trait that have been mutated. And this procedure is called complementation or a complementation analysis. So let the two mutant strains be A1 and A2. And we want to determine whether A1 and A2 are two alleles of the same gene, or whether A1 is a mutant allele of one gene and A2 is the mutant allele of a distinct gene. And in these analyses, we always assume that the wild type allele is dominant and the mutant allele is recessive, which turns out to be generally the case. And to do complementation, it's quite simple. What you do is you cross the two strains, you cross the two lines, and you will get different outcomes depending on whether these are different genes or whether it's two alleles of the same gene. So let's discuss the case, the first case, where we have A1 and A2 are mutant alleles of different genes. And let the wild type alleles be called A1 plus and A2 plus, and the mutant allele be called A1 and A2. And now if these are two different genes, then we can discuss two alternative scenarios. One is that the genes are not linked, so they are on separate chromosomes, and the other scenario is where the genes are linked. So let's do the first situation where the two genes are unlinked. Now, since we have a pure breeding strain of the A1 line, or the A1 allele, they must be homozygous for A1, and they also must have the wild type allele of the other gene, so they should be A2 plus over A2 plus. Similarly, if you look at the mutant strain that is A2, since it's pure breeding, it must be homozygous for A2, while it must be homozygous for the wild type allele of the first gene. And when you carry out this cross, we will get all individuals hets between the wild type and mutant allele of A1 and hets between the wild type allele of A2 and the mutant allele of A2. And all the individuals are going to have this genotype. Now, since the mutant alleles are recessive or the wild type alleles are dominant, all the progeny of this cross, when we cross the two mutant strains, are going to be wild type. So what we've done is we have taken two strains, two individuals who had a mutant phenotype, and crossed them, and instead of getting mutant progeny, we are getting wild type progeny. And that's why these two genes are set to compensate for each other's defects, or in other words, they are set to complement each other. Now, let's discuss the second scenario where you have A1 and A2 as mutant alleles of two different genes. And that is when A1 and A2 are linked on the same chromosome. Once again, the first line is mutant for A1 and it's pure breeding, and therefore it has it's homozygous for the mutant allele, while it must have the wild type allele of A2, since it's very unlikely that when we get a mutant, it will be actually mutant for two different genes at the same time. Similarly, the second line is homozygous for the mutant allele of the second gene, A2, and it must be homozygous for the wild type allele of the first gene. Now, when we carry out this cross, all the progeny are going to be A1, A2 plus over A1 plus A2. And once again, all the progeny are heterozygous for the wild type and mutant alleles of both the genes. And since the wild type alleles are dominant, which is usually the case, all the progeny are once again going to be wild type, and the two mutant strains are set to complement each other. Therefore, if these two strains that we have isolated are actually mutant for two different genes, crossing the two strains together will give us all wild type progeny, and because the genes will complement each other, and we know then that these are, in fact, we are dealing with two different genes that control the same phenotype or trait. Now let's discuss the last possibility, which is, what if the two strains that we have isolated are actually mutant for a single gene, but we just have two different mutant alleles of the same gene. In this case, when we take a pure breeding line mutant for A1, it will have two alleles of A1, and the other line will have two alleles of A2. And when we cross these, all the progeny are going to be a het between A1 and A2. And this is also called a trans heterozygote, where you have two different mutant alleles in the same individual. But the main point is that since there is not a single wild type allele, and you have both the mutant alleles, all the progeny are now going to be mutant. And in this case, then, the two strains do not complement each other. And we know then that these are two mutant alleles of the same gene. The two strains that we have identified are in fact the same gene that has been mutated in two different ways. Let us discuss complementation with the example of the harebell plant. Normal wild type harebell plants have blue flowers, but three mutant strains have been isolated. There's the dollar strain, the pound sterling strain, and the yen strain, all of which give white flowers instead of blue. And our goal is to determine the dominance relationships between these genes, whether these strains are mutant for one gene, two genes, three genes, and so on. So the first thing one would do is one would cross the wild type with each of the mutant strains. And when we do that, we get all blue in the F1 generation and a 3:1 ratio of blue is to white in the F2 generation. And what that tells us is that wild type is dominant. In the next step of the analysis, we perform complementation, where we take two mutant strains and cross them and look at the phenotype of the progeny. So when we cross the dollar and yen strains, we get all blue or all wild type individuals, and that means that dollar and yen alleles, they complement, which from our preceding discussion implies that they are separate genes. When you cross the pound and the yen strains, then we again get all blue or all wild type progeny, and that implies again that pound and yen are separate genes. They complement each other. Finally, when we cross the dollar and the pound strains, we get all white or all mutant progeny, and therefore dollar and pound don't complement, since we did not get all wild type, and that means dollar and pound are mutant alleles of the same gene. And what this implies is that we have a total of two genes, let's say W1, and the wild type allele of W1 we'll write as W1 plus, which has two mutant alleles, W1 dollar and W1 pound. So these are the mutants. Whereas we have a second gene, let's say W2 plus. So the wild type allele of the second gene is W2 plus, and the mutant allele is W2 yen. So when we cross the dollar and the pound strains, so we're crossing dollar and pound, we are in fact crossing a pure breeding strain dollar, so it's W1 dollar over W1 dollar, with the other pure breeding strain, which would be W1 pound over W1 pound, and all progeny are going to be trans heterozygotes, $W1 over W1 pound. In other words, they will all be mutant, and that's why they don't complement. On the other hand, if we are crossing, let's say, dollar and yen, then and assuming that these genes are unlinked, we are in fact crossing W1 dollar over W1 dollar with the wild type allele for the second gene, so W2 plus over W2 plus, and this individual is the dollar strain, and the yen strain is wild type for the first gene, so W1 plus over W1 plus. Let me make some more space over here. And the second strain, the yen strain, is homozygous for the mutant allele of the second gene, so W2 yen over W2 yen. And all the progeny are going to be W1 plus over W1 dollar, so they're going to be heterozygotes for the first gene as also for the second gene, so W2 plus over W2 yen. And since the wild type is dominant, the wild type alleles are dominant, as we saw previously when we crossed the wild type strain to all the mutant strains and got all wild type progeny. All the progeny here are going to be blue, or they're all going to be wild type. It turns out that the reason for this particular behavior of complementation in hairbred plants is because the W1 gene, the wild type allele of the W1 gene, makes an enzyme that converts a colorless precursor into a second colorless precursor, whereas the W2 gene, or the wild type allele of the W2 gene, makes an enzyme that converts the second colorless precursor into blue pigment. If in the dollar mutant, you are W1 dollar over W1 dollar, and therefore this step is blocked because there is no wild type allele W1 plus to make the enzyme, and therefore you are stuck at the first colorless precursor stage, and the plant is the flower is white. On the other hand, if you are a pound mutant, which is a mutant allele of the W1 gene, you are W1 pound over W1 pound. And once again, there is no W1 plus allele to make the enzyme, and you are again blocked at colorless precursor 1, and you are a white flower. If you are the yen strain, then you are W2 yen over W2 yen, and you do have functional you're homozygous for the wild type allele of W1, and therefore the first colorless precursor gets converted to the second colorless precursor, but it cannot get converted into the blue pigment since there is no W2 plus wild type allele of the W2 gene that makes the enzyme to convert this colorless precursor into the blue pigment, and therefore you are again white. Now, when you cross the dollar strain to the pound strain, you are W1 dollar over W1 pound, and you lack the enzyme because you don't have any W1 plus alleles, and you are blocked, and you can never make the second colorless precursor for it to get converted into blue pigment, and you end up as a mutant or white flower. Finally, when you cross the pound strain to the yen strain, then you are W1 plus over W1 pound and W2 plus over W2 yen. So you have one wild type allele for both W1 and W2, and that means you have both the enzymes, and a colorless precursor gets converted to colorless precursor 2, which gets converted to blue pigment, and W1 and W2 complement each other. Finally, you should practice with this problem. In minks, wild types have an almost black coat. Breeders have developed many pure lines of color variants for the mink coat industry. Two such pure lines are platinum, blue-gray, and Aleutian, steel-gray. These lines were used in crosses with the following results. Are the platinum and Aleutian lines mutant for the same gene or different genes?